semi auto analyzer চলো বেড়েছে হ্যাঁ তাহলে আমরা AST প্রোগ্রাম কি কেমন হবে what aspartate Aspartate Transaminase. Aspartate Transaminase. এই এনজাইমটা কি করেন এটা পুরো নাম আছে কি নাম SGOT মানে কি oxaloacetate কি করে বল ইঞ্জিনের নিচে এনজাইমের রিঅ্যাকশন করে তো কি হয়েছে জোরে বল অ্যাসপার্টেট থেকে আচ্ছা প্লাস আলফা কিটোগ্লুটারেট আচ্ছা আমি যদি দেখি কিভাবে লিখি আমি যদি কিভাবে লিখি কিভাবে লিখি তাই তো রেডিও অ্যাসপার্টেট কি হবে আলফা-ketoglutarate কি হবে কিনেটিক হ্যাঁ পুরো বলি তুমি আর এই রিঅ্যাকশনে এটা ইউনিভার্সাল এনজাইম লাগে কি নাম বাহ কত বলেছে তুই সেকেন্ড ঠিক আছে বাহ ঠিক সাপ থাকলে বলিস এখানে সাপ আসে আসে এখানে থাকে আমি থাকে সাপ আসে আর একটা মিশাপ আছে পোশাক ওয়াটার মিশাপ তো মানে বুঝতে আছে যদি এইটা অ্যাসপার্টেট থাকে তাহলে আমি যদি এসি যদি অ্যাসপার্টেটের জায়গায় বলি এই দুটো এনজাইম আমাদের সবাই বাংলা থেকে বাংলা গার্লফ্রেন্ড কি বাঙালি বয়ফ্রেন্ড নেই ওর মা নাকি মহারাষ্ট্র Anwar is Dhan is Bab. Both are Bab. Dhan is Bangali, Bab is Bangali. Dhan is Bab. What do you want? Good. So these two enzymes are always found in all tissues. In all tissues. Mark my word. They are found in all tissues. What is the equation to use here? In. Now tell me why it is available in serum or plasma. Serum or plasma is not in tissue, it is outside tissue. So we are measuring. First point is we are measuring in serum. Why? যখন আমি বললাম এটা টিস্যু মধ্যে পাওয়া যায় টিস্যুতে নামার পর বাড়ে কেন হচ্ছে এই প্রশ্ন বলছি ডিজিজ কন্ডিশনে হয় আমি যে নর্মাল গুলো দিচ্ছি তখন নর্মাল গুলো ডিজিজ নেই আরে ঠিক বলে কারেক্ট বলছে 90% কারেক্ট Endpoint কি কারেক্ট করে নাও কারেক্ট কত বুঝি নম্বর কারেক্ট আসলে ও আলোচনায় থাকি দুই তাই তুমি কত দিস বুঝিস বল আমি বললাম যে AST আর ALT এর টিস্যুর মধ্যে পাওয়া যায় কিন্তু মাপছি আমরা সিরামে টিস্যু বাইরে কেন পাওয়া যায় নর্মাল আমি পাওয়া যায় Here, hey, English কতদিন বলছি Here, you have to remember the term called non-functional. What are non-functional enzymes? Non-functional enzymes are found in plasma or serum, and these enzymes are called non-functional because first of all, non-functional enzymes are found in plasma or serum, and they are called non-functional because their natural substrate is not present in serum. In the result, the instrument power substrates are absent in serum or plasma. তাদেরকে বলা হয় non-functional. Right? Now these non-functional enzymes are found in plasma even in low disease condition, small amount. In some disease condition it increases, but with no disease or normal physiology, some amount is also present in the plasma serum. What is this amount for, and why it increases during certain diseases? When certain disease occurs, then the tissue gets affected, the tissue is damaged, so the cell membrane is destroyed, the tissue. So the enzyme is poured outside the tissue. Where? Plasma. But whereas in normal physiology, when you have normal person with no disease, some or other cell is always dying. Normally, those cells will contribute to this normal level of enzymes. It's true for amylase, lipase, alkaline phosphatase, AST, ALT, everything. Correct? So these enzymes are called, all enzymes are called non-functional enzymes. What are the functional enzymes present in plasma and serum? Next question comes. Although that is not a topic to be taught in this class, but just for your information, if you are interested, you can keep in mind the enzymes which perform in clotting action. Clotting the coagulation of cells require functional enzymes. Correct? So basically, we are measuring some non-functional enzyme, the plasma serum, using kinetic method. Now, enzymes are measured what is mass? The quantity. What is the unit? Unit mass is measured. Sit down. Right? Speed. Right? Velocity. Right? How to measure enzyme activity? How fast or how slow the enzyme is doing? If we say like this, is that right? Is that right? Okay or not? Water, not that much height. অক্সিজেন ধরে যাবে ঠিক আছে এই জায়গায় করা বেটার এখানে চারটি রিঅ্যাকশন পাচ্ছি আমরা চারটি রিঅ্যাকশন four reactions, four constant. এবার এই কনস্ট্যান্ট আমরা জানি কি জানি k2 by k1 plus k minus 1 is what? K case special case m minus 1. Here, suppose this particular enzyme, what we are doing? We are measuring aspartate right? So what is the major problem here to measure? Which one to choose as substrate? Take a substrate, write a substrate. First question. So both sides let's see. What is the answer? আমি S মাপবো, P মাপবো, T মাপবো. There are three quantities. One is E, S or speed, right? These we cannot measure. Okay? Can you measure? You know what about? I'm here to my arm level. So I'm going to try to do it. You know what I do? The data is here. One. It's here. Even though there is actual factor, but কোনো reaction, in some reactions, it is easier to measure substrate. In some reactions, it is easier to measure product. Okay? It is never easy to measure enzyme. Why? Because by the laws of any reaction catalyzed by a catalysis, whether it is biological or non-biological, chemical. Right? Enzyme remains unchanged after reaction. So why to measure enzyme? Right? So we have to measure either substrate or product. So here, what is the substrate and what is the product? That is the question. What we are doing? Can we measure this one? No. Why? Because the other substance is also present in good amount in the blood. How do you know when the reaction started and when it ended? So, first, the hurdle is to determine whether we should measure speed or velocity. What is the difference between speed and velocity? How do you measure? Suppose a boy is driving a motorcycle in a circular path. Yes. Right? Velocity will be different every time. This way, this way, this way, this way. Right? And then the speed is uniform. So what we should measure? First hurdle is, should we wait the reaction to end and then we measure the total amount of product formed, because initially product was 0. At the end of the reaction, product will be something which will form due to conversion of substrate. So that is called endpoint method. Okay? And when this is not possible or cumbersome, then we measure speed. How? From point A to point B, we will measure, suppose 30 seconds or 1 minute. So 1 minute, how much substrate is converted? We measure. If we measure substrate, then the concentration will be negative because with time our substrate will be decreasing. It will be minus AC. If we measure product, then it will be so now we have two options. We can either measure substrate, increasing concentration in the first minute and in second minute. So in the first minute it is 10 gram per second, suppose. In the second minute it is 12 gram per second. So now we add and divide by 2, what we get 11 gram per second. So the speed will be at least 11 gram per second. Okay? Now how much is required? How much substrate is being converted? We measure, then that will give what? A value. When you say how it will give a value? How it will be the actual value of substrate when we are measuring the kinetic method. We are seeing only a window. We are not going to the end of the reaction. How much is total substrate is being converted? No. That is not equal. That is not equal. Because the total amount of substrate is somehow related to what? How many substrate molecules we participate in the reaction per second. If suppose there are 1000 molecules of substrate, right? So in first second there will be 10 utilized, second there will be 12 utilized. Second minute, divide 11 molecules will be utilized per minute. Right? So if we see if it is 1000, then it is 11, we cannot measure. But if it is 20, then measure. Right? So we take huge amount of substrate which will not suddenly collapse. If we go for endpoint estimation, if we take small amount of substrate, we go for kinetic estimation. Because we see only one window. This is not the then we dilute the sample because it is more than the amount can be measured. We dilute sample, run the same test. Dilute sample further, run the same. It should be increasing and decreasing. Then only it can measure it. Right? So if you are suppose measuring 2500 units per liter AST, that means how much AST you have to be, how much substrate you have to be, if it is endpoint. Almost a bag full of substrate. But if you do the kinetic method, you have to give less amount of substrate. It is more cheaper. So there are two methods. One is kinetic, another is endpoint. In endpoint, we end the reaction completely and then we measure the total amount of substrate converted. In kinetic, we measure the amount of substrate converted against unit time. Okay? Now we know one molecule of enzyme, one mole of enzyme will react with one molecule of substrate to produce one mole of product. That equation we have already reduced. If it is 2, the equation will be different. If it is 1, then it is this second order reaction. If it is 1, it is second order reaction. If it is only 1, first order reaction. If it is 0 order reaction, which calculation we perform in the class? zero reaction. No reaction takes place zero order. This is imaginary. Without substrate, without product, no reaction will take place. Because the simplest way to calculate will be that. Anyway. Now you know what is called kinetic method and what is called endpoint. Okay? Next. Now suppose we decided to perform it in kinetic method. Good. Kinetic method. Let's start. Now the next question is which one to measure after this four? All are regularly found in plasma. Cannot calculate which molecule was previously present and which molecule is converted to oxaloacetate. No. What will we do? We will take very small amount of serum. How much? 10 micrometer. 20 micrometer, 5 micrometer. It can be very small amount of already formed oxaloacetate. But again, that much small amount of oxaloacetate measurement is also difficult because that will give a reaction, a running reaction. You cannot measure a flying bird. It's always difficult to kill a flying bird. Why? Because it's always changing its point. You cannot aim and shoot. So we take the other thing which is not present in normally in the blood. What is that? We take oxaloacetate. And make it here to be NADH plus H plus to form NADH. Now, tell me, do you find NADH normally in plasma or serum? Yes or no? Answer is no, already I told. Why? Well, why? Why we don't get NADH normally in plasma? Hey, last bench. Or how's that student? Or we will pass it down. NADH is impossible to find in plasma or serum. Why? Because plasma or serum both contains heavy amount of water. NADH being a energy-rich compound cannot survive or sustain in aqueous medium. What do you think? Good. So then we conjugate another reaction with this. Now we have very small amount of oxaloacetate formed, already formed, and through that mostly this aspartate, what is converted to oxaloacetate will be highest amount, correct? And that highest amount is there. So oxaloate concentration will be contributed mostly by the oxidized, converted from aspartate, not which was previous replacement. Because that will be small. And this reaction, this NADH and NAD, this conversion, we can measure NADH, but we cannot measure NADH. We can measure NADH in UV range, that is 340 nanometer. 340. What is visible range? What is 340 is below. It is UV range. Infrared. UV. Also, so in the UV light, so can we see the color? Unlike the previous tests what we have done, urea, creatinine, glucose, total protein, they were showing colors. Why? Because they were 540. So no color change. In the UV range only, this NADH concentration will be highest in the initial reaction, and it will decrease with time. Right? So if we draw our 2 points, then we can draw a straight line. If we have more points, then we can draw more points, but we have only 2 points. One is after 1 minute, another is after 2 minutes. So after 1 minute, reagent, this is where we started, comes here. After 2 minutes, it comes here. So this portion of the graph only shows the linearity. This is a lower value, this is a higher value. So what is the linearity? For every reaction, this company people have measured their linearity range and they have provided in the book. Right? Within this linearity only we can measure, not beyond these points. If the values come beyond these 2 points, we cannot measure. Right? For that, we have to bring the result within this range. How? Suppose you are given a job to measure the distance between bottom 1 and pole 2. How do you measure? With a foot scale? The foot scale, then laser device, arc. In the arc, globe is already there. Draw a line, then measure the latitude longitude, where this is. By road, if you come, you measure by laser line from where to where it falls. Right? Why? Why not for food scale? Because then you have to tie to measure in food. So what we, for seeing the quantity, we change the scale. Suppose we have very less amount of AST. We come below this value. So undetectable. If it is very high, then it goes beyond this point. It goes beyond this point. Then we dilute and bring that concentration within this range, then measure. So that always we measure within these 2 points. This is the basic understanding of how a kinetic method works over an endpoint. And if we endpoint, then we have only one time of incubation. So 2 minutes, 3 minutes, like you have in this up, goes after 5 minutes, after 10 minutes. whatever amount of glucose is there, it will be converted, and that reaction will sustain because of that amount of glucose is fixed. Here, AST can have very high value. How long will it do it? It will be an hour. That is impossible. That's why to deal with such a huge amount of entities, we use mostly kinetic. Okay? So basically we have learned each one. AST, what is AST? Why AST? Why AST we will learn later. Why, what is kinetic method and why kinetic method here? And lastly, using water analyzer. That has made our life very easy. We don't have to calculate any further manually. Now, like improve solve. The lead is what? Reaction. So now, this is the part I will not because I will just tell you to take this method down. Let me just give this heading first. Estimation of serum AST by kinetic method using serum oxaloacetate using same oxaloacetate. Then first comes what? Principle. The principle of reaction. Okay. This you can take photograph also. I don't mind. Finish fuse buffer pH 7.8. Why pH 7.8? Because to maintain this reaction. If it is 6.5, then the reaction is opposite. So we make it alkaline, so the reaction from this side goes to this side, that side. It's better. Change the pH. So if it is only 10 micrometer of sample variance, so and to that we are adding almost 1 mole of reagent. So a lot of reagent, small amount of sample. So if you overpower the reagent sample, now I am able to support it. Aquí यहाँ बुझी माने किया उठला उठला now the composition of fuse buffer, the composition buffer is fuse buffer, L-aspartate, NADH, NADH means married dehydrogenase, right? Lactate dehydrogenase and stabilizer. Okay. Now everything see what is the amount of aspartate presence 240 millimoles per liter. Small. If it is endpoint, then you require large amount. 6.8 substrate. What is there in substrate? This is present. This is we have to provide. So we provide alpha-ketoglutarate, and then we provide NADH, and then stabilizer. Now these two have to be mixed, region 1 and region 2. Before using. Why before using should be mixed? Because NADH is highly susceptible. So if NADH we mix beforehand, then it will get destroyed. It should be mixed just before the test. Right? This will be mixed just before the test. Fine. Now third is apparatus. I have written as before. So what you have written will you be what the apparatus required for any estimation by Serum analyzer? Number one Serum analyzer. Number 2, pipettes. Number 3, reaction tube or reactive. Right? Number 4, water bath. This is what was not required. Why? Because the kinetic method is already fed into the semi-organizer, so it will incubate within the instrument. You don't have to do anything, just mix it and feed it immediately. There we have to wait for after incubation, endpoint. In hybrid, we have to just mix and immediately we have to feed into the instrument so that if you take inside, it's aspirate the sample along with the reagent and it will incubate within the instrument and give us a result. Okay. So apparatus as before, so write this apparatus. Number 4 is the procedure. Just write the table form. How much sample, how much reagent is added. See, 100 microliter of sample or serum or plasma and working AST reagent 1 ml. Right? So we require pipette for 1 ml, we require pipette for 100 microliter. Then mix well and aspirate immediately for measurement. Suppose you can read absorbance after 60 seconds, leave there, you can write because we don't have to do this manually. Instrument will do it. And then read directly delta A is change in absorbance. So you will have A0, A1, A2. First time first interval for A1 minus A0. Second interval is A2 minus A1. Right? এবার 0 যোগ করব, বিভাগ করব, mean বেরোবে, you take the mean as a difference. So delta A by mean. Then you go to number 5. The calculation part, you just write, you don't have to worry about what is called what. Because kinetic factor is something very difficult to deduct. But scientists have done on our behalf and they have taken a small amount of charge against which they have purchased this reagent and doing the business. Right? So they have calculated and they have made it. So I have just made it mark with first bracket C. Okay. One was the factor is that is if you want to convert it into the SI. Then number 6 comes reference value for male and female almost similar 30 less than 35, 5 to 35, 30 to 40 normal. Then number 7 is the result. Whatever result you get, during examination you have to write only what? Principal and result. So principal you have to read it now for all conditions, all the parameters. Okay. What is the basic principle? And then result. And what you have added. Procedure means what you have added, the table form. What is the table form? This table form. Pretty easy. How much you are adding to what? This one you write procedure and then result. That's it. You don't need to write composition of reagent, apparatus, only. Give me example. Okay? Principle is required. Why? Because that is on what basis we are estimating how great. Right. Now we come to the clinical correlation of AST. What do we get after estimation of AST? We get after estimation of AST the exact value of AST in either SI unit, or the local unit. There are different units of enzyme activity to measure. One is given here. See the number 5, last line. AST activity in nanocat in cat by liter is equal to AST activity in international unit, which is SI unit, international unit, IU per liter into 16.67. This is conversion factor. Right. For example, for blood glucose, what is the normal level? Suppose 90. milligram per deciliter. What is the SI unit for that? Specialty. Milligram per deciliter means what? You convert it into first liter. Then you divide that milligram amount by the molecular weight of the glucose. It becomes millimoles. All molecular glucose molecule. So divide, you get 3.5 to 5.6 millimoles per liter. It is same. Similarly here also it is same. Now what we measure for AST? AST we measure for particularly two diseases. One is liver disease, another is myocardial infarction. That too, it is not one of the among the earliest marker of myocardial infarction. It is a late marker of myocardial infarction. So the person comes to you after 1 day or 2 days having chest pain. Then you will not find CK, you will not find myoglobin, you will not find troponin I or T. You will find the AST and LDH elevated. AST and LDH elevated. Why? Because they are late markers. They start to increase late, after 24 hours. Okay. So AST is elevated, but liver is normal. That means that AST is due to myocardial infarction. Why? Because patient is also the complaint of chest pain 2 days back. But that time he was growing the harvest, so he didn't have time. He took rest, and then after growing the harvest, then he has come to see the doctor. Then you see AST is elevated. Moreover, you find changes in ECG also. In blood, I am mentioning. ECG, I am not mentioning. Right? So, there you find AST is elevated. In liver disease, if it is deep liver disease, then both AST and ALT will be elevated. Chest pain, AST, ALT both are elevated. Non-conclusive. Can't say. What is it? AST elevated, ALT normal, chest pain history, MI. Chest pain, AST, ALT both are elevated. Liver, nothing to do with chest. Can't say. Because it is too late to conclude for MI. But you see other parameters, other tests, ECG, echo. Right? So like this. So AST level is very, very important for two purposes. One is MI, which is a late marker of MI. Another is liver disease. Now, what are liver diseases? Which type of disease? Hepatic jaundice. That is viral jaundice. Right? So the hepatic jaundice or viral jaundice will show increase in AST elevated work. Can they decrease? They can drastically decrease below 5, 1 or 2, 0.5, 0.2. When at the end stage of cirrhosis of liver. Initially during cirrhosis of liver, these factors, both the factors increase, AST and ALT. But at the end stage, the patient is going to die bigger or more. Then nothing is left in the liver to produce any normal AST ALT. All the liver cell has been damaged. Can we have zero urea? the end stage of cancer. The liver is totally destroyed, compromised. It cannot produce any urea anymore. The urea level will be very low, 1 or 2. Just 2 days back I got one. I inquired, you know what? Got the news that patient is suffering from cancer. So since these enzymes are mostly produced by the liver and heart, what will this AST? So if liver disease occurs, this increase, heart disease occurs, this increase. But for heart, it will increase late, after 24 hours. By that time, most of the early markers will come to normal level. Only AST and LDH will be high. So that's which LDH? LD1. LDH1. How do you differentiate LDH1 from total LDH? You measure LDH1 and total LDH. This is the difference. Which is more elevated. Any question? और खाता में लिखा है ये खाता में लिख के ला अगले दिन मंडे कल वेडनेसडे ये प्रैक्टिकल होगा प्रैक्टिकल